Thanks so much, Lisa. I'm so excited to be here. We have a lot of very interesting things to discuss with lots of very interesting people. As we're celebrating our 30th anniversary, I can't help but reminisce with our first researcher, as Defeat Duchenne Canada has a long history with Dr. Rudnicki, who is among the first grant recipients we'll speak with, and who was one of the first ever grant recipients from Defeat Duchenne back in 2006. Dr. Michael Rudnicki is senior scientist at the Regenerative Medicine of Ottawa Hospital Research Institute, the director of Regenerative Medicine Program and Sprott Center for Stem Cell Research, also at the Ottawa Hospital Research Institute, professor in the Faculty of Medicine at University of Ottawa, and scientific director of the Stem Cell Network. Dr. Rudnicki, we are proud to have first supported your research in 2006, like I said, in our very early days of funding. That grant was to provide equipment for analysis of gene expression in candidate stem cell populations, which would result in enhanced understanding of the genes that regulate stem cell function and characterize promising cell lines and insights into the biology of muscle regeneration. You can tell that I just read that because I wanted to make sure I got it right. So at the time of our grant announcement, we made the statement, in the future, identification of gene targets has the potential to lead the development of drugs that would ameliorate the progression of muscular dystrophy by stimulating the inherent regeneration of capacity of skeletal muscle. We've come a long way. In 2025, there are several treatments available, although not currently approved in Canada. And we have several more in development, including gene therapies, small molecules, such as developed with Satellus Bioscience, which you are obviously contributing to developing. Back in 2006, would you have imagined the level of progress that we've made in this time?
Oh, it's really snowballing, I would say. It's even more than snowballing. It's an avalanche of new discoveries. The science has really progressed incredibly rapidly, and the numbers of clinical trials that are occurring is very exciting. And the way forward looks, I think, very positive.
Where can you see us in 30 years from now with
Well,
Duchenne?
I think many of us working in this field imagine that we'll have a cocktail of drugs and approaches and interventions that will dramatically change the course of the disease. I don't want to use the C word, but it's conceivable that with a cocktail type approach that it will change from a lethal disease to a chronic disease. That's our aspiration, and 30 years seems a very reasonable timeline for that to happen. I don't want to raise false hope, but I think it can happen faster.
That's wonderful. Well, I'll hand it over to you now to give us an update on your current research grant, which was awarded in 2022.
Okay.
So take it away. Thanks so much.
Let me share my slides. So we're getting towards the end of someone else is sharing. So I have to replace current share and PowerPoint. Here we go. And okay, am I sharing the full screen? I think so. Is that right?
Yep, it's perfect. Thank you.
Okay, good. And you can see my cursor.
Mm-hmm.
So this is a project that's we're approaching we're in the last year of funding, I think, and we've really made very nice progress. We're working on a secreted protein that we discovered that's released by muscle when it's been damaged, and it's called Wnt7a. And this protein has remarkable properties, which I detail in this slide in a number of publications that we've put out. It drives the expansion of muscle stem cells. It stimulates the migration of the blast cells that repair the muscle, their dispersal, and it also stimulates the growth of muscle fibers. And we found that if we delivered it to a dystrophic muscle, it made the muscle stronger and the damage much less acute. So it has a very high potential as a therapy for Duchenne muscular dystrophy, especially to treat the muscle loss that occurs. But the problem with Wnt7a is that if you inject it into the blood system, it doesn't go anywhere. It's so sticky, it remains where you injected it. So we can inject it directly into muscle, but we can't deliver it throughout the body. But we subsequently discovered, and this is the subject of the grant, that Wnt7a is not secreted as free protein, like when we're injecting it as free protein, but rather it's secreted from cells in these little vesicles called exosomes. And in these little vesicles, these little packets, it can be delivered through the blood system and go long distances to reach other cells that can react to it. Um, uh, and so we were really interested in understanding that process and studying it. So we did a number of things. First, we developed a process to purify these exosomes carrying Wnt7a. And there you can see Wnt7a sitting, those little black dots sitting on a vesicle, the exosome. This process is scalable and can be used in good manufacturing practice type facilities to make clinical grade material. And we published this method just recently, a few months ago. Um, uh, Secondly, we did a really interesting piece of work. So this is what Wnt7a looks like, its structure. It looks like a clamp, and that clamp grabs the end of the receptor that's on the surface of proteins and binds it and activates signaling downstream of that receptor. But what we did is we identified a signal peptide that we termed the exosomal binding protein that acts like a postal code to direct the protein to go to the exosomes and traffic to be packaged with the exosomes and be displayed on the surface of the exosomes. And this sequence is a very short 18 amino acid fragment. We can link it to other proteins, and they also go to the same place. We showed that this protein binds another protein that's on the surface of exosomes called COP-B2. And here's the structure of the two together. So it's a really neat biochemical molecular study showing how Wnt7a can be directed to and secreted on exosomes. And that was published just recently. And more importantly, we can take exosomes and we can deliver them into the blood system. They travel throughout the body and they make muscles stronger. You can see this is an increase in force generation. This is a muscle that was damaged in an acute injury using cardiotoxin in the leg. And we can show that it goes, dystrophic muscle becomes quite a bit stronger when we do this single injection of this material. And also undamaged muscles, and this is the diaphragm, when we inject EVs that lack Wnt7a, you can see the dystrophic changes in the diaphragm are not affected. Whereas in the mouse that received these EVs, we have really a much healthier looking muscle with areas of damage much reduced. So it appears as if regeneration has occurred. And this has also been published recently. So we've shown that Wnt7a is secreted on exosomes. We've identified the postal code sequence that can direct it to exosomes. We figured out how that mechanism works. And we can also take that postal code, link it to something else, and have it go onto the surface of exosomes. And that's important because we want to enhance its targeting and get more to it, to muscle. And very importantly, we can deliver this through the blood system. So right now we're doing a mini clinical trial in dystrophic mice to treat repeatedly with exosomes and attempt to generate experimental data that would be proof of therapy. Below are the people who did the work in my lab. Thank you.
Wonderful. Thank you so much for that update. We really appreciate hearing all about your research.